just wanted to do a brief introduction. I am Babette Henderson. I am the Senior Director of Alumni Engagement for the Feinberg School of Medicine, and I'd like to welcome you to our virtual event with Dr. Amy Paller. Before we get started, I wanted to cover a couple housekeeping items. If you'd like to ask a question of us or of the presenters after the presentation, please utilize the Q&A chat feature below. Please use that instead of the raised hand, as our moderator will be posing the questions to the panelists. We encourage you to use speaker view versus gallery view to view the meeting, and you will be muted throughout the meeting. And if you have an unstable internet connection, you can increase your speed and connectability by turning off any robust applications or internet connections on your other devices. Now I'd like to introduce our moderator for the event, Ashley Cousins. She's an Associate Director in the Office of Development of the Feinberg School of Medicine, and she will introduce our panelists and moderate our Q&A session after the presentation. Ashley?
Hello, thank you all for joining us. Dr. Amy Paller is the Walter J. Hamlin Chair and Professor of Dermatology, Professor of Pediatrics, and Principal Investigator of the NIH-funded Skin Biology and Diseases Resource-Based Center at Northwestern. She received her undergraduate and graduate degrees from Brown University and her medical degree from Stanford University. Dr. Paller completed residency training in both pediatrics and dermatology at Northwestern University and her postdoctoral research fellowship at the University of North Carolina. She served as Chief of Pediatric Dermatology at Northwestern's Children's Hospital until she became the Chair of Dermatology at Northwestern in 2004. An author of almost 500 peer-reviewed publications, Dr. Paller has also edited several major textbooks in dermatology, among them Hurwitz's Clinical Pediatric Dermatology, 6th edition recently completed, and Fitzpatrick's Dermatology and General Medicine. She is an NIH-funded investigator in both her laboratory-based and clinical research. She has directed the Pediatric Dermatology Clinical Trials Unit at Northwestern Lurie Children's for the past 25 years and has been the lead investigator on several landmark trials in pediatric skin disease related to atopic dermatitis, psoriasis, and other skin disorders in children. Her laboratory focuses on skin innervation in inflammatory diseases and topically delivered gene regulation through nanotherapy, including for psoriasis. Without further ado, Amy Paller.
Thank you so much, Ashley. Well, I want to welcome all of you today. There's so much to talk about with respect to dermatology at Northwestern, but what I've chosen to talk about is how we've reached out to others to bring in technology to do dermatology research. And with that, I'll tell you a little bit about our department and then show you some examples of that. So dermatology has been consistently ranked either number 2 to number 5 in the United States in NIH funding. And part of that is because we have established here a skin disease research center. It's now called the Skin Biology and Diseases Resource-Based Center. This is NIH funded, and we are now in our 11th year of 15. That's pretty much unprecedented that we've had this for 3 full cycles. But it has facilitated an incredible amount of collaborative research here at Northwestern. We have 74 faculty members who utilize the SBDRC from 14 different departments. And in our latest rendition, we have 3 novel service cores. One is called the STEM core, in which we developed 3D models that look very much like skin, and it allows us to do all kinds of manipulation to test and then be able to look at changes that we see morphologically. We have a new unit that's called the Test It unit that has a component that does particularly immunologic tests, all kinds of things, helping people who would not otherwise have them in their laboratory. And we have another unit that allows us to bring in people who have perfectly normal skin, who are willing to volunteer to help us either get a sample or test ultraviolet light or some other way to perturb skin. And then we also have individuals who have skin problems like eczema or psoriasis, who volunteer to come in and might receive a special treatment but then might be biopsied to do some of the technologically driven studies that I'll be mentioning today. And finally, we have a core that allows us to change cells by getting in genetic changes and see what happens. We also are very fortunate to have the largest dermatology clinical trials unit in the country here at Northwestern, and that includes both pediatric and adult focused trials. We currently have, actually, I think it's closer to 200 now, active IRB approved projects, about 90 of which are clinical trials, but the other are all investigator initiated. And that means a lot of new trials that we're driving ourselves, or new investigations that we're driving ourselves. And that's led itself to an infrastructure of 26 full-time people who just are involved in clinical research, supporting about 20 clinical dermatologists who do research. Now you can imagine the interplay as well between the SBDRC and the clinical trials unit. We've really facilitated getting a lot of samples from patients that's allowed us to do human-based research, highly translational. Now, the other thing that I want to mention is that skin is an amazing organ for doing research. It is the largest organ, and it's also the most accessible organ. That means we can use it for delivery of medications. That means we can use it for understanding the biology of the human body, and particularly of skin. And that also means that we can use it for monitoring. And in all these various areas, we've pulled in technology just to do that. Now, I don't have to remind you how common skin disorders are. There are probably very few of you who don't have something on your skin that's not perfectly normal. And sometimes these can be life-altering and disfiguring. So I've listed here some of the skin disorders that are the greatest burdens to society, whether we're talking about cancers or inflammatory or infectious or just tumors of other sorts. There are just so many. Now, I do want to mention that we're always trying to drive collaborations because that's what drives good science. And I think there's nothing more important, and I speak as a physician scientist, than really taking the bedside to the bench, to the laboratory, and then figuring something out that allows you to go back into the clinical arena and make a difference in how people are treated. So a program that we fairly recently started is a small grants program, and it's really funded through the generosity of donors. So I did want to mention it today because I'm hoping some of you might be interested enough in investing in some of this. This is a new program that we developed to foster clinical and translational research on skin diseases that specifically gets our clinicians to merge a program with a scientist, can be in dermatology, can be outside, can leverage our SBDRC cores because they're really successful in supporting people, but it allows biopsies, or as you'll see, other ways to get material from humans to be able to be studied using technology. And we are giving funds for up to $10,000 to any of these combined clinician scientist pairs for a total of $50,000 a year. Really excited about this program. Now, I'm going to start by telling you about delivery. So we know that delivering treatment topically can be advantageous. It concentrates medicine at a site where the skin problem is, and that makes it more effective, but it also avoids potential risks of taking medicine internally, side effects, interactions, so it's safer. And it's often cost effective because you don't need as much to deliver something topically as to get it in to the skin through the entire body. Now, most topical treatments that we have, unfortunately, are pretty nonspecific. Think about steroid creams. People are worried about using steroids on their skin. Most of the time, that's not really reasonable, but nevertheless, the fear is there because we know that there can be nonspecific side effects. So we want the most targeted treatment possible. And that's where we're going right now with topical therapy. We're understanding the cause of disease and using that to develop new pills, new shots, but also ideally targeted therapy. And there's been much less targeted topical therapy. Now, why is that? It's because skin plays an important role as being a formidable barrier. It prevents water loss, so we don't dry out. It prevents loss of other things, keeps everything in, but it also keeps things out. So it's keeping out pathogens like bacteria and viruses, it's keeping out things that are allergic triggers or irritants, but it's also keeping out potential medications. So you have to be very small to get through skin. Our skin is like a wall of bricks that are the cells and mortar, which is fats between the layers. And it's very important that if we want to deliver something topically, we've got to figure out ways to get through this barrier. It's a huge challenge. It's like the holy grail for topical medication. So one of the things that we did, and this actually was a collaboration that started more than 10 years ago with this faculty member, a guy named Chad Merkin, who is part of the chemistry program here and is the head of the International Institute of Technology that's based here, nanotechnology that's based here at Northwestern. Now, he was telling us that he was able to do something that we didn't believe, and I'll tell you a little bit about it. He was awarded one of our very first pilot studies from the Skin Disease Research Center as a result. He said that he could take a nanoparticle, put genetic material on it, get it into any cell, and knock down genes and change what's happening in those cells. And we were able to team with him and show about 100% uptake into cells, including skin cells, which are really, really hard to get anything into. But then we said, well, what is this? And can we actually get it into skin itself? Now, it turns out these are called spherical nucleic acids, and they are nanoconjugates, meaning that this is nanotechnology with a conjugate to genetic material. And the trick of it is in a dense spherical array. And now it's come in all different flavors. It can have a gold nanoparticle core. It can have something called a lysosomal nanoparticle core, which is essentially lipids. Sometimes it's just what's on the outside itself. And what we found over time is that we can put it in a common moisturizer, rub it on the skin, and it actually gets in. So what you're seeing at the right is something, these nanoparticles labeled with a red fluorescent dye that shows you that it's getting in. And what we can see here, this is the outer surface of the skin. This is the outer layer or epidermis of skin. You can see all the cell nuclei in blue, and you can see a lot of red because when we put it on the surface, we're able to get it in. And if you look even deeper below the outer layers of skin, you can see that it's actually even getting into deeper layers. So we were able to show that it gets in. It didn't require anything special physically like a laser or other type of techniques. It didn't require any special chemical agent to penetrate. It was because of this dense spherical array of genetic material around a central nanoparticle or just the nanoparticle type shape of it itself. It's very, very small. Now, we found other factors about it, like it wasn't easily degraded. There was no evidence of toxicity. We looked in human skin, in mouse skin, in cultured cells. And then we started applying it. I'm just going to briefly mention some of the research that's moving forward on this. Now, one of the areas of research is related to diabetes, type 2 diabetes, very, very common problem. And unfortunately, many of these individuals get these chronic non-healing ulcers, especially if the diabetes is not in control. Therapy's largely been ineffective, and many end up having amputation as a result. So we're able to work with mouse models. This is a diet-induced obese mouse. I think you can see that the mouse is a little chunky, and that's because they eat high-calorie stuff. And we use that as a way to do our studies with diabetes. Now, we made a discovery a while back that knocking out a gene that makes something called GM3 prevents these mice from developing diabetes. And we've been able to show using these nanoparticles, these spherical nucleic acids directed against something that makes this GM3, that we're able to very quickly heal in these diabetic mice in a way that we're not able to do in in those that are untreated. So here we have an untreated, we have another with a fake SNA, and then we got with our particular with our particular nanoparticle down here, and this is just in a similar, but in a free form that's not in the nanoparticle configuration. And I think if you look at day 12, you can see a dramatic difference. And that's what we've seen with these nanoparticles. Now, we've also taken them into another common problem called psoriasis. I'm sure everybody here knows of somebody who's had psoriasis, because 1 to 3% of the population gets this. We see this in children, we see it more in teenagers, and we particularly see it in adults. And we understand what drives psoriasis. It's a part of the immune system that involves too much of something called interleukin-17, too much of something called TNF. And we also know that we've had in the last 10 to 15 years a whole bunch of new therapies, which are called biologics, which involve subcutaneously injecting antibodies that target the various pathways that we've discovered cause this psoriasis. But unfortunately, they're too big to get into skin. So none of this has been relevant for those who would want to use a topical product. And frankly, that's 80% of individuals with psoriasis who have more mild to moderate disease and otherwise are just using potent steroids. So here we have another mouse model. Here we can put something called a miquimod on the back of the mouse, and we can induce something that clinically, that under the microscope, looking at tissue, and also looking on a molecular level, looking at the very smallest particles, looks like psoriasis, including these increases in IL-17, these increases in TNF that we're talking about. And we've been able to use this as a way to test whether these topically delivered spherical nucleic acids targeting either TNF or targeting interleukin-17 as a pathway can make a difference. So we treat to make them get psoriasis, and we treat to see if we can prevent it. And what you're seeing here is in fact just that. The one on the left here shows you when we target TNF, which we know is really causative as a factor in psoriasis, that as opposed to those that are controls, shown here in the graph in the upper lines, those that were treated with the spherical nucleic acid have something that really in the square where we were applying it looks virtually normal compared to the surrounding psoriasis-like skin. And we can also use this to target the interleukin-17 as another one. And I think you can look and see here in these mice treated with controls, this is without anything inducing the psoriasis. You get dramatic improvement with this, with the other target as well. And we can similarly see in green the ones who were treated with this target versus the control ones. So using it for diabetes, using it for psoriasis, and the psoriasis ones have already gone to humans, already completed phase 1 trials. Now, I'm just going to give you one other example of how we've partnered with our nanotechnologists here at Northwestern to try to use technology towards treating human disease. And I talked a little bit about skin, and these can be used for skin too, but I'm going to just show you, even in dermatology, we try to be diverse. We've got scientists who work on both skin and eye as another surface. And this is Shad Thaxton with Robert Lavker. Robert's in dermatology, Shad's in urology, and is a nanotechnologist. And Shad's invented these natural high-density lipoproteins. Now, I think everybody knows what high-density lipoproteins are. When we look at our cholesterol levels, we like having high-density lipoproteins. We don't like having the low-density lipoproteins, which give us high cholesterol for the most part. But he's been able to replicate these with a gold core nanoparticle into something that looks like a high-density lipoprotein. He's actually using it, trying to use it for cardiovascular disease as well. But what we found is that these are able to enter skin and eye cells, that they're non-toxic, and that they can transport genetic material, just like I was talking with you before about, or proteins into cells. And so they're able to penetrate even in intact skin and eyes. And I want to just show you as another tickler a little bit about developing eye drops for diabetes. So here they're able to show that if they induce wounds in a diabetic-like model, that is the mice like I showed you, that are given a diet that makes them fat and diabetic with type 2 diabetes, here we're able to see that in those that had the particles themselves, we have markedly increased closure of the wounds that are induced in the diabetic eye as opposed to the slow closure of wounds. We know that's a problem in diabetes. It's a problem in the eye too. And you can see that here we have the diabetic mouse eye not treated with anything but a control. We have a perfectly normal eye healing and something in between with this other form of nanotherapy. And that's moving towards more trials to try to see if this can move forward therapeutically. I'm going to shift gears now towards the second area, understanding human biology. And that is including on a personal basis. And here we have really been leveraging some of the incredible technology that's been developed around the world that we have at Northwestern in our core center and certainly in other cores at the university to try to understand the biology of skin. I'm going to use an example, something you may never have heard of. This is a condition called ichthyosis. It's a group of genetic disorders characterized by red scaling skin, usually everywhere on the body. Ichthyosis is from the Greek for fish because of all the scaling that these individuals have, and they often are born with this and they have it lifelong. And unfortunately, there's not good therapy for this. And we don't understand much, although we've come to understand the underlying genetic changes that occur that cause this. But why are these patients so red and scaly? What is it between the very beginning with a genetic change in all the cells in their body and when we see redness and scaling? And this is so important for us to understand, because if we can understand better all of the various stages that get to redness and scaling, we can interrupt something along the way without doing gene therapy, for example, and make a big difference in their outcome. And if you're red and scaly your whole life, you can imagine they're itchy, they're uncomfortable, and they're stigmatized because of the highly visible nature of their skin problem. So our idea was to try to understand, and maybe even with some of the newer therapies coming out for more common problems, be able to repurpose a medication that might be out for this group as a whole, if we can just figure out what makes them red and scaly. And the beauty of technology right now is that you can take a single 4-millimeter biopsy, that's about the size of a pencil eraser, very small, and you can take a blood sample and you can find out a tremendous amount about what's going on in the skin from that biopsy and what's going on systemically from looking in the blood. And we've been doing these studies in this group with ichthyosis for the last few years, and you can see a series of papers starting in 21 patients, then 29, then 47. Now we have more than 60 patients involved in this research. And what we've been able to show is that from that little biopsy and from blood samples, we actually were able to define what's going on in the immune system in skin and blood that can give us some clues for treatment. Now, I know you probably are saying, what is this? This is called a gene array, and it's laid out in such a way, first of all, on the left, to look at this group of patients with ichthyosis and compare them to two common problems. One being eczema or atopic dermatitis, the other being psoriasis, both common inflammatory skin diseases with some scale. And in fact, both of them have new treatments available that we were thinking we might be able to repurpose for this group of genetic disorders for which there's no therapy. And I think you can see colors and notice the blue normal, the red at the top for psoriasis, the largely blue that looks more normal with atopic dermatitis, and then red for the column that's labeled ichthyosis. And I will tell you that that's what's shown in the Venn diagram on the left, where you see a large number of genes that are increased or decreased, increase shown in red, decrease shown in blue, overlapping not with eczema, but overlapping with psoriasis. as you see here with the color coding on the right. And there are some examples of psoriasis. I showed you one before, another that has a lot more scale, a red scaly disorder. And this is RNA-seq. It's yet another technology tool that allows us to look with a small amount of what's called RNA extracted from the biopsy, and we can look at over 32,000 genes here. And it turns out that the top genes, the ones that are increased the most, and here you see a variety of different forms of ichthyosis, all increase in these top 30 genes are indeed some of the top 30 genes that we also see of the immune system that are increased in psoriasis. So we've shown here through this research that this group of genetic disorders shares a psoriasis-like pattern with too much of the arm of the immune system that I talked about before, with that IL-17, where I showed you in those nanotherapy studies. It's called the Th17 arm of the immune system. So the question that then comes up is, does this excess of the immune system that's skewed towards what's called Th17 drive the inflammation and scaling of ichthyosis as it does for psoriasis? And if that's so, could we possibly repurpose some of these medications used for psoriasis to treat ichthyosis? And I'm going to show you some examples that start to say yes. So here's a little girl, she's 9 at the time, and she has a form of ichthyosis that's also associated with inflammation of the heart. And she was very early on in infancy, first came in to see me with what we thought looked like psoriasis actually, but it wasn't. Later went on to have what looked more like an ichthyosis. And at 5, she went on to have heart inflammation. She had to get a heart transplant. You can see the scar going down the front of her. You can also see that she's red and scaly because of this form of ichthyosis. Now, what we were able to find is that, in fact, when we did just those kinds of studies, we found what was increased. We're able to put her on a special medication that's used normally for psoriasis. And we also put another boy from Ireland with the same form of ichthyosis on that medication. And in both cases, within a few months, it was life-changing. Do you see how she seems to have big cheeks? She seems to be a little puffy. That's a sign of someone who takes systemic steroids. She, in fact, had a heart transplant, but she wasn't taking steroids. She was using anti-inflammatory medications that weren't doing a thing for her skin, but she was using topical steroids twice daily because she was so terribly itchy. I think if you look at her before and her after, you can see that not only is she no longer red and scaly, but her face looks so much thinner. Because 2 weeks after she started this new medicine, she was able to stop this topical steroids. She was able to stop having a situation where, because she was so red and had such a poor barrier to her skin, it was just being sucked inside and treating her as if she was on them systemically. So both she and this boy did well. It was life-changing. We're now 3 years down the road, and they still look like they have mildly dry skin and are doing very well. Here are some other reports in other forms of ichthyosis. This is a boy as a toddler, red scaly, now almost 9 years old, gets shots every 8 weeks, doing very well. A 20-year-old with monthly injection. This just came out, 4 cases, including this boy who had another form of ichthyosis, showing the dramatic improvement. And there are now studies in adults and children that are either ongoing and planned. I think this is going to be highly effective if we treat early enough. And in fact, we're about to start a study in children with the drug that was used here with one of these cases. Now, biomarker analysis is what's this called, where we use big technology to look for biomarkers, things that are increased, things that are decreased, and that are associated with disease. And we can use this as well, not just for rare disorders like the group of ichthyoses, but more common skin disorders as well. So we've worked on eczema and have used just this type of profiling to show that even though the work to understand it has been primarily in adults, there are some different profiles if we look at young babies in the first 6 months of their disease versus older children versus adolescents versus adults. And in other studies, these types of technologies have shown differences based on racial and ethnic groups and even individual by individual. But everything I've talked about to be able to look at skin involves a biopsy. Let me tell you, I'm a pediatric specialist. Skin biopsies are really tough to do in young children. They're just not feasible for studies in children, and adults don't like them either. So we've switched to something in children called tape strips. These are painless, they're not scarring, and yet it's a way to get an individual's profile that's feasible enough to do its serial visits in clinical trials. This is just a little bit about the tape strip technique. We stick it on, it's like a big circle of tape. We can do 16 tape strips in a child. We can do 20 tape strips easily in an adult. We use a device, we take it off, and here you can see we're collecting skin, and this is skin from the outer layers, but it seems to be enough for us to be able to do these studies. I showed you before a picture. This is again looking at RNA, and you can see that as opposed to the normal controls, we can see that even what looks like normal skin in somebody who's a baby with eczema is not normal. We've known that for a long time, and we can see even greater changes in the lesional skin. It's important to let you know that of these 16 tape strips taken from all these babies, 99% yielded enough skin to run our analyses. 97% of the markers we tried to look at were detected. And we even did RNA-seq with more than 32,000 genes. And we saw the same kind of a profile that we saw in the biopsies in these tape strips. So we think they can be used. It's now been shown for adults with eczema, adults with psoriasis, even to use for melanoma. We're now looking at the tape strip we collected from patients with ichthyosis to compare them from the biopsies, but we're pretty convinced that this will work. And we're planning studies with many other skin disorders, including for the first time in children, because we can do this non-invasively. So what are we able to do with this? Well, we talked about RNA, looking at genetic material that tells you what's being produced on a genetic basis, but we can look at proteins using something called proteomics. We can look at the lipids that are critical to cell function. That's called lipidomics, metabolic alterations, metabolomics, and we can also look at the microbiome. That is what bacteria or fungi or viruses are on the skin and are altered in disease. And all of these give big data that's then processed through something called bioinformatics. This is not only helping us to find new therapies, but it's ultimately going to lead to personalized therapy, because we can start trying to understand how to predict responses, how to predict disease course. I want to also mention another technology, artificial intelligence. Dermatologists, of course, what we're good at is recognizing patterns on skin. What is the color? What is the shape? What is the location, etc.? And from that, we diagnose things. The question is, can a computer be taught to do the same with help from dermatologists? And at Northwestern, we do this through what's called augmented intelligence. It's artificial intelligence, but it's really working hand in hand with the physician, not replacing the physician, but working with the physician so that the physicians can do a better job. Now, we've already been doing studies in the last 2 years. We worked with Google to get all kinds of pictures, and Google's working with us to look at skin lesions to diagnose cancer, inflammatory skin problems, etc. But we're also working internally. We have a new institute here at Northwestern called the Institute for Augmented Intelligence in Medicine that's led here by Abel Coe. I'm showing his picture here. And an example of one of the grants we just put in that's going to get funded is for the genetic disease on the right. This is called epidermolysis bullosa, probably the worst possible disease you can imagine. These patients all over their bodies and internally have blisters that lead to scars. They're in chronic pain, chronic itch. They get progressive deformities from the scarring and go on eventually to get squamous cell cancers of the skin that are so severe that they metastasize, and they usually lead to death in these individuals who've survived this life that's just horrible. So we find that these are very hard to diagnose. We find that people don't want to come in for biopsies. This is such a depressing disorder for them. And we're trying to use artificial intelligence, or particularly augmented intelligence, working with the Institute here to try to figure out how individuals at home can take pictures of these, and we can use these to know when to bring them in for biopsy, and as early as possible recognize these metastatic squamous cell cancers that, if caught early, have a much different outcome. Finally, I want to just talk about the technology of monitoring through skin using sensors. And some of you may recognize this is some of the work that's been put out and really publicized extensively from John Rogers and his group through the Center for Biointegrated Electronics. And we've played a huge role in dermatology through that. I started work with John several years ago, actually, on wound healing, but we teamed together to try to work with monitoring using these sensors wirelessly in neonates, as I'll show you in a minute. I pulled in Dr. Steve Zhu. Now, Steve was a medical student at Harvard. He came to Northwestern to do his dermatology residency with a background as an entrepreneur and also a background in engineering. This is exactly what he wanted to do. So Steve ended up doing fellowship work with John Rogers, and now is a faculty member who is the medical director of the Center for Biointegrated Electronics, and is really taking it to an incredibly high level, working collaboratively with John Rogers. So I think many of you are aware of some of the work that we introduced into the neonatal intensive care unit here at Northwestern, with the idea that these babies have so many wires, it's hard to get to them, and it's very hard not only for doctors and healthcare workers, but very importantly for their parents to be able to hold them and cuddle with them. So our idea of the future was to have little devices that are tiny skin-like sensors that would attach to them and would be doing all of this monitoring wirelessly and using technology. And as you know, this has been highly successful. This was our first paper where the three of us were the senior authors on this science paper, changing the monitoring in the neonatal intensive care unit. This was a huge project with 45 co-authors, and it's just gone on from there. We've had a few things since that time. There's Dr. Zhu in the neonatal nursery, 250 neonates to date, and we have 2 wireless sensors. One goes on the chest, and the other, as you can see now, uses this device that just wraps around the foot and is able to monitor consistent temperature and blood pressure, which before were only intermittent, as well as all of the other parameters, looking at their heart rate, their respiratory rate, etc., including how well they're oxygenated. And we can use this in fragile premature skin because we're not using very tight adhesives. And this is the type of thing it can read out on any kind of a mobile device. We don't even need the fancy machines that are right now used to read out the wired devices. We got a lot of press coverage for this. It was unbelievable when this first came out, and huge value actually for Northwestern, which was fantastic as well. In fact, this was the most viewed Facebook video post in Northwestern's history, with almost 200,000 views, about no wires, more cuddles, which was the goal here. And I like to boast that second place was Northwestern University's basketball team, making the NCAA tournament in 2017 with less than half of those views. So really got a lot of press. And this has moved on not just for further advancements in the neonatal ICU, but in many spheres. I just want to mention fetal monitoring. You can see these small devices, very unobtrusive on the mom. And this is the program that's been taken with this very flexible small device to deployment in India and Pakistan and Zambia and Kenya, with funding from the Gates Foundation and Save the Children. And I will tell you the count is now at 70 children being monitored in Kenya and is growing. A few other things to mention. This is a cool device that just collects sweat on the surface and can analyze through an automated system glucose and other ions, and so that we can start to look at metabolism. You can imagine how it could be connected with something that could then sense and deliver for diabetes. And we can use it for other things just by collecting sweat as a non-invasive way to look at something rather than a blood collection. This is one of my favorites that I'm working on. If you don't recognize it, this is a child with that very itchy disease, eczema. And this kid is incredibly itchy, and we're trying to treat with all kinds of topicals to make him better, but he's quite uncomfortable. And I asked the team to make a scratch sensor, and this is what they came up with. This is called the ATOM, the Advanced Acoustico-Mechanic Sensor for Scratch Detection. It's a flexible device that captures the motion and sound of scratching. So it's way beyond what could be replicated even with the actigraphy, which is a wrist bracelet, which is far better, by the way, than any Fitbit or other such device. And this is how it works. This is a little model we have with eczema. You can see she's got a little device on her chest. She's got a little device on her dominant hand. And we can capture all the vital signs from this, but we can also capture what's happening with her. Let's see. I don't know that the sound's working. Sorry about that, but usually we can hear the scratch, and we certainly can see the motion when she's scratching. This is an infrared sensor in the studies that we're doing to see the correlation and the likelihood of a scratch. Very, very high correlation. So they can sense scratch versus movement versus rubbing with this kind of a device. Now, this whole technology finally it's really been taken as well into COVID-19. Here's a device that's been introduced by the team with the expectation of having 500 subjects by the end of 2020. It's a wearable technology, like I have been showing you, sits imperceptibly over the throat with something the size of a Band-Aid, and then captures the three most important early signs of COVID, that should be COVID-19, fever, shortness of breath, and coughing. It counts the coughs and the cough intensity and senses labored breathing. It can distinguish from allergies and colds and dry throat. And because it's close to the carotid, it can also monitor the heart rate and the blood pressure. So this is something that's been rolled out for individuals at risk to try to monitor on a regular basis and see, is something happening, to catch it early and get them in. I'm very proud of these various technologies that are going on in dermatology. I want to just mention 2 dermatology department scientists were among 12 global finalists for Nature Medicine's spin-off prize that honors academic entrepreneurs who have developed spin-off companies that have a global impact. Now, one of them is the Cibel Health. This was founded by Drs. Zhu and Rogers based on the work on neonates at Northwestern. And another is Dr. Carolyn LaPoule, a key part of our Skin Biology and Diseases Resource-Based Center, who studies gene-based treatment for vitiligo, which won a prize as one of the 12 finalists. I think you probably know somebody with vitiligo, easier to see in darker skin, but it's total loss of pigment in certain areas and can be highly problematic for patients who develop it, and there is no treatment. I will tell you that of these 12 global finalists, very proud that the winner, the top prize, went to Dr. Zhu and Rogers' work on our neonates at Northwestern. So in conclusion, that large body surface area and easy accessibility of the skin makes it ideal as a focus for the collaborative research that we do here at Northwestern that leverages our skin-based center. That merging the strengths of clinician and scientists with these technologies using our cores at the medical school, including the SBDRC, and with faculty experts, for example, in chemistry or engineering, we're using technology to make discoveries towards personalized medicine, towards new targeted therapies, and for devices that are going to measure skin health and disease. And these are just some of the ways here that Northwestern Dermatology is changing the world. And with that, I want to thank you for your interest. I know we're not going to have a ton of time, but feel free to contact me at any time. That's my email address, and I'm happy now to close this and take any questions that you may have. Thanks for your interest.
Thank you, Dr. Poller. That was a fantastic presentation. I'm going to start asking some of the questions we've received. The first one, are there gender differences in skin diseases and disorders?
Yeah, absolutely. And gender differences are now really a high priority at the National Institutes of Health. In fact, there are very few studies that we're doing now that aren't trying to look at gender differences, and we're finding them. So, of course, we know about gender differences in certain diseases being more common in women or in men. The whole COVID-19, much more common to have disease and severe disease in men, for example. In collagen vascular disorders, rheumatic disorders like lupus, for example, much more common to occur in females. And we're also seeing different responses. For example, one of our skin scientists has discovered that the responses in skin to steroids and topical steroids are different in males versus females, and is trying to understand that. And we're looking at male versus females in terms of other kind of responses and risks by doing this very research. And we can look at that by taking samples from females, taking samples from males, and doing these types of studies comparatively.
Great, thank you. Our next question, we received a couple questions regarding augmented intelligence. And do you see this ever fully replacing the work of the dermatologist, or, you know, in the next 20 years, what is the proliferation of artificial intelligence and machine learning in terms of diagnosing and recommending treatments?
I'm very excited about augmented intelligence. I'm sure hoping it's not going to replace physicians. I think physicians will always play a role, and I hope we never get to a point where that important connection between a patient and a physician is central to healing. But what we're looking for are ways that the doctor can do the job more easily. There are already some technologies out there that use some augmented intelligence, or use some of the techniques even I showed you today, to try to get some information, to try to help triage, to sort out which patient should be seen or not. I'm excited about the work we've done with Google. I'm really excited about some of the work that we're doing now internally moving forward. I think you can imagine that if we can start much more rapidly heading towards diagnoses, or figuring out which patients really need to come in for biopsies or other diagnostic techniques that take what is suspicious and turn that into a yes/no answer, it's going to make our job that much easier.
Great, thank you. We've also had a couple questions come in regarding teledermatology and what concerns you might have about it, especially in terms of the ethics.
Well, you know, teledermatology has really revolutionized how we saw patients during the pandemic. We've done teledermatology for years on a pilot basis. Suddenly we rallied into pretty much doing it exclusively for a few months until we were able to open up again. We're still doing some teledermatology. It doesn't replace somebody coming in, but it's a good substitute for some people. Many people live far from experts, and it's very hard for them to get the medical care that they need. That can be in the United States, it can be around the world. There are also patients who probably don't need to come in. They are being seen in follow-up. They're doing well on a medication. I will say there are limitations. For one thing, there's the whole price structure with teledermatology. One of the reasons we couldn't do much before is because nobody was paying for it. And it does take a lot of time. I'll be perfectly honest. Teledermatology with a patient can take me twice as long as seeing them in the office setting, because it can be complicated. And you're talking a lot, and there's a lot more questions. And for me in dermatology, I can never see what I want to see well enough through the video of teledermatology. I have all my patients send me pictures that they take in advance, so I'm also looking at them and using good images with good resolution in a way that I can't really see as well usually with teledermatology. And that's why some of our store and forward can even work, which means basically they're sending pictures, filling out a form, asking questions, I'm giving answers, may not even be at the same time. So I think there's an important role for teledermatology, because for certain things it's great. I think it's still very challenging and risky. For example, if somebody has a pigmented lesion that may be melanoma or some other form of cancer, and you are relying strictly even on a picture, because now we have other techniques like dermoscopy, total body imaging. For example, here at Northwestern, we have something called IntelliStudio, where in 4 minutes, one of our patients will go in the room, and we have this incredible camera that takes full body images, high resolution, in 4 minutes. Now, we could never do that with telemedicine, of course. So I think that there is the ethics involved with who's paying for it, who's not, and there's always a system where if people can't afford it, maybe they can't do telemedicine. There's also the problem, though, that I worry about, that if we depend too much on telemedicine, we're not able to look in a way at doing full body skin exams or even looking closely at what might be cancers, for example, and do a good job on that. So I still, even with relatively easier ones, for example, the acne patient in follow-up, or the eczema or psoriasis patient who's doing well, and I have my pictures attached, I still like to see them every so often. So I don't think it'll take over, but we certainly are thinking of ways to make it easier for patients to augment our care with them through use of teledermatology.
Makes a lot of sense. Thank you. We have a question about what ways can technology support rural and underserved communities?
Well, I think part of that is the same thing I was just talking about. So rural, underserved communities where access is harder is a great opportunity to use telemedicine in general and teledermatology as well. And also for the patient population that's somewhat disabled. I showed you a picture of some legs that were quite deformed and covered with scars and blisters. So we have a center for excellence here for these patients who have this rare genetic blistering disease. And it's hard for them to get into, see the doctor because of their disabilities. And I've done some of my patient visits through having them take total body pictures with dressing changes, and then have what could be quite a long visit with them, going over what I'm seeing and what their concerns are, and I'm able to see them in that way. It's the same thing with rural, it's the same thing with underserved areas in general, that we can do a lot of this, and we can triage through them. And there are going to be patients in whom you say, I'm not able to do a quality visit here, I'm going to need for you to come in, and then they have to travel to that specialist. I think it's also an opportunity, though, for primary care physicians to better work together with specialists to do teledermatology. I'm hoping in the future that what we've learned and what we're learning now with the COVID-19 pandemic will allow us to do inpatient consults in some of the many Northwestern medicine hospitals where they're needed, but there hasn't been a dermatologist to service that. We can have our hospitalist team here downtown take care of all this if we can get up systems that work and have decent resolution, and we just work it all out. I'm hoping that we can have more where primary care physicians, or even in some of our immediate care facilities, can be having a system that leverages our ability to do this teledermatology, to provide consultation or to provide some quick answers back within 24 hours in an urgent care setting, these types of things. So I think there are many opportunities to leverage this judiciously, wisely, and to the advantage of many patients.
Sure, absolutely. It looks like we have time for just one more question. This last question, in your view, how should the future of technology trainees look, and what should they expect in the clinical landscape and training to change as a function of current technological developments?
Well, I can't even imagine, even 5 years from now, certainly not 10 and 20 years from now, what our trainees will be learning, but I can guarantee you that it will be incorporating so much of the technology that I just talked about and that we've been discussing. There's no question that teledermatology is going to get better and better and is going to absolutely be a part of that. There's no question that augmented intelligence will be a part of that. There's no question that we're going to have different diagnostic techniques that are so far beyond what we're able to do right now, that's leveraging some of these technologies, whether it's taking that tape strip and suddenly looking at 30,000 genes, or the large number of proteins or lipids or metabolic signals, or looking at the microbiome. I can just tell you, I haven't published this yet, but we took different types of patients with ichthyosis, another center for excellence we have here at Northwestern, and we just did swabs, and we took the DNA from those to look for bacteria. And I can now, because of data I just got back, tell you who has what subtype of ichthyosis just by the bacteria on their skin. We are just starting to scratch the surface to understand what this big data will do. And there's no question that we are going to be leveraging that in 10 years and certainly in 20 years to make decisions about what that patient's course is going to be, what comorbidities, what other disorders may develop because of their risk factors, and how to pick the best medicine early on for treating that disease, including that skin disease.
Thank you. All right, well, I think that wraps up our questions for now. Thank you so much for hosting this wonderful presentation. Thank you for everyone who's joined us, and we will be sending out a follow-up email so that you have access to a recording. And there will also be information linked there as well if you would like to learn more about ways to support the Department of Dermatology. I hope that you all have a great evening.
Thank you so much.